Alright, so if we're discussing cells, one of the things that we need to know is how we make more. Cells are constantly getting damaged. If you get a bruise, alright, you've damaged some blood vessels and the cells of those blood vessels. If you cut yourself, alright, you've damaged the skin cells and you need new ones. And so cells are constantly reproducing and making new cells. It's also how we grow bigger. You know, you're born, you're only this big, and when you're full grown, you're this big. And the only way to get bigger is to add more cells. And so the cell cycle is how cells actually work and then reproduce and create new cells. All right, so this is the sequence of growth and division of a cell. There are two general periods in the cell cycle. All right, the first one is called interphase. Now, interphase is the growth period of the cell. Literally, the cell's getting bigger. All right, and it is during this stage that a cell grows in size and carries on metabolism. And what that basically means is it's just doing its work. That's what it means to carry on metabolism. The cell is doing the work that it needs to do, whatever its job is in the body, it's doing that job. All right, and that all happens during interphase. Now, very important for making new cells is that during interphase, chromosomes are duplicated to prepare for division. So remember, your DNA is found inside your nucleus, and the DNA tells your cell what it does and why it does it. All right, and so if you're going to make a new cell, you got to have a copy of the DNA in the new cell. And so you duplicate the chromosomes so that when the cell actually divides, each cell will have the DNA that it needs to know what to do and how to run. The second general phase is called mitosis. Mitosis is the actual period of division. So when the cell actually splits, okay, it's when the cell's nucleus and then its cytoplasm actually divide. And so eventually the cell membrane will actually divide also, of course, to get two daughter cells. And that's just what we call them, as daughter cells. All right, so when you look at the whole thing, it is a constant cycle. That's why they draw it as a circle. You'll see that most of a cell's life is interphase. All right, all of this is interphase. It's broken into different periods, this G1S and G2. You don't have to know that, I just wanted to show it to you. But so the cell is growing, the DNA is being replicated, so it's copying all of its chromosomes. It grows some more and gets ready to divide, and then it goes through mitosis. This is when the cell actually divides, and this is what we're going to be talking about for the most part, is this actual division here, when you actually get two new cells. All right, so let's look at those phases of mitosis. Now remember, interphase, the cell is growing, it copies its DNA, it does its job, and then it divides. And that division has 4 different phases. So that's what we're talking about now, is the phases of mitosis. Mitosis is divided into 4 phases. They are prophase, metaphase, anaphase, and telophase. And you can use the abbreviation PMAT. So PMAT, P-M-A-T, prophase, metaphase, anaphase, telophase. All right? And this is a little memory device to help you remember. If you remember that the P, prophase, P stands for prepare. Because in prophase, the cell is going to prepare itself to divide. And then you have M, that's metaphase. And think of M standing for middle, because that's when the chromosomes are going to line up in the middle. We'll talk about that some more in a minute. And if you think about A, for anaphase, A equals apart. That's because that's when the chromosomes are pulled apart. And then if you think of T, telophase, T standing for 2, that's when you finally actually get 2 new cells, is in telophase. All right, so this is just to help you remember what happens. In prophase, the cell prepares. In metaphase, everything moves to the middle. In anaphase, everything pulls apart. And in telophase, you finally get two. So let's look at what is actually happening in those phases. All right, in prophase, chromatin, long tangled spaghetti-like strands of DNA, coil up into chromosomes. And at this point, remember, they've already made a copy. The copy was made during interphase. And so each duplicated chromosome has two halves. And they're called sister chromatids. And they are exact copies of each other, completely, totally exact copies. And they're attached to each other, all right? They're actually stuck together here, all right? And those are the sister chromatids. Also in prophase, the nuclear envelope and the nucleolus begin to disappear. Basically, you'll end up with a cell with no nucleus because it just kind of breaks down so that this can happen. All right? And then the centrosomes, which are these small cylindrical structures made of microtubules. They're called centrosomes. They begin to migrate to opposite ends of the cell. So literally, you have this big cell and everything's kind of in the middle. Well, one centrosome moves to one side and one centrosome moves to the other side. So opposite ends. All right, oops, sorry. And as they migrate, they start forming a spindle between them. Now, these are representing your centrosomes. These little things that make it look kind of like spiders, those are the spindle fibers forming. They're going to make a spindle. All right. The nuclear envelope here is drawn dotted to show you that it's starting to break apart. All right, and these would be your sister chromatids attached to each other. So one chromosome and its exact copy that was made, and they're attached. So they're joined up, as you can see here, okay? So those are your coiled up chromosomes, and they're attached at the center. All right, now the spindle, what these little fibers are going to be making, the spindle is a football-shaped cage-like structure. It ends up basically looking kind of like a oval bird cage. All right, and it's going to play a part. Oops, I did it again. It plays a vital role in separating these sister chromatids from each other. All right, so that all happens in prophase, and then next is metaphase. And so here you can see the spindle. All right. And you can see that spindle fibers have attached to these sister chromatids. There's one to each half. All right? And so this is kind of what it looks like. And remember I said that M stands for middle. Look where these things are lined up. Right in the middle. All right? And that's why M stands for middle. Because the chromosomes attach to the spindle fibers, okay, by their centromeres. That's what these little orange circles are representing, are the centromeres. Okay, and that's the center of the chromosomes. And then they're pushed and pulled in whatever direction they need to go to line up on the midline of the spindle. So they'll end up being in the middle of the cell. So that's why we say that M stands for middle, because that's when all these sister chromatids get lined up in the middle. All right, and that's all that's happening in metaphase. And then each sister chromatid is attached to its own spindle fiber. I'd already said that, sorry. All right. And then in anaphase, remember A for apart, and notice what's happened here. The sister chromatids have been separated and they're apart from each other. That's why we say that A stands for apart. All right. Anaphase begins with the separation of the sister chromatids. All right, and they are pushed and pulled apart toward opposite ends of the cell, or opposite poles of the cell. All right, and so now your identical copies of your chromosomes that had been attached to each other have been pulled apart from each other, and they're at opposite ends. All right, so each chromatid is now a separate chromosome because they've been separated. All right, and that brings us to telophase. And telophase begins as the chromosomes reach opposite ends. So as soon as they get up there at those opposite ends, okay, telophase begins. the spindle is going to break down, so that cage is going to disappear. The nucleolus will reappear, and a nuclear envelope will form around each set of chromosomes. So for just a minute, you have a big cell with two nuclei in it. A nucleus at one end and a nucleus at the other end, because that nuclear envelope forms back up. All right? The chromosomes then unwind. So they've been all nice and coiled up. Now they unwind and they become that big old tangled mess again. But they have to unwind so that they can direct activities of the cell. So now that they're unwound, they can tell cells what to do again. All right? And then finally, the plasma membrane begins to separate the two new nuclei, the nucleus at each end. They're separating them to make two new cells. All right? And so what happens is you get all these chromosomes at each end. Now the nucleus starts coming back. That's what this little dotted, looks kind of like a Mike and Ike's to me, start showing back up. All right? The spindle is disappearing, and you're going to have a nucleus at each end. All right? And so when you put it all together, this is kind of what it looks like. So in prophase, everything starts breaking down. You have your sister chromatids attached to each other. In metaphase, there's no nucleus. You have a full spindle here, and you have them lined up in the middle, sister chromatids lined in the middle. Anaphase, they have been separated and they're moving to opposite ends, so apart from each other. All right, then you get to telophase, and you have a nucleus at one end and a nucleus at the other end, and you're starting to separate the plasma membrane. See how it's starting to pinch in right here? That's going to separate it until you have two brand new cells. And these two brand new cells are back in interphase. Okay? These are going to uncoil. You won't actually be able to see chromosomes when you're in interphase, but they just left them in this picture so that you could see that you had the halves. Okay? Now, when that cell actually separates, so you have a nucleus at one end, a nucleus at the other, and the cytoplasm has to divide so you can create two new cells, that's called cytokinesis. So the cytoplasm dividing is cytokinesis, and that's how we get two new cells. And it works a little different between an animal cell and a plant cell. In animals, the plasma membrane actually pinches in along the equator to form two new cells. And that's called a cleavage furrow. Now, if you think about it, a plasma membrane is soft, it's flexible. And so if you squeeze it, you can actually pinch it together and then pull it apart. And that's basically what's happening in an animal cell. It squeezes in on this cleavage furrow right here in the middle until it totally pinches in half and it separates. Okay, that's how animal cells separate. But if you remember, plants have a cell wall. And cell walls are hard and rigid. They're literally like a wall. All right? And so you can't squeeze that. If you squeeze a wall, it crumbles. All right? And so you have to separate a plant cell a different way. And you do that by basically building a new wall in the middle. All right? So because of that rigid cell wall, a structure known as a cell plate forms across the equator. The cell plate is basically a new wall being built right down the middle of the cell. So the cell plate is built right in the middle, and that's going to separate the two new cells. All right. The importance of mitosis is that it guarantees genetic continuity, all right, by making two new cells identical to the old. And that's important. If you cut yourself and you need some new skin cells, well, when you grow those new cells through mitosis, you want the new cells to do the exact same job as your old cells did. Okay? You don't want stomach cells growing out of your arm. You want new skin cells. All right? And by doing mitosis and copying that DNA and giving each new cell the exact same DNA, the cells will always be doing the same job. So that genetic continuity allows the new cells to do the same job as the old cells did. So that's why it's really important. All right,